All right, ladies and gentlemen, welcome back. So today we're going to talk a little bit more specifically about how meiosis and sexual reproduction really are able to achieve that genetic variation in offspring. And I think this is probably one of the coolest components to what we talk about when we talk about cell division is just how do I get this genetic variation? Why don't I look identical to my parents? Why don't I look identical to my sister? Right? And it comes down to the way that that genetic information gets passed down. So when we talk about meiosis and the process itself and what's happening to the genetic information, the DNA, as it's going through that process, that genetic variation comes from really two sources within the process of meiosis itself. And that's from independent assortment and from crossing over. This leads to all sorts of genetic recombination. We're going to take the information that your parents passed on to you, mix it all up, and then stick it into your gametes, your egg or your sperm, before you then pass it on to your own children. So this is that huge advantage to meiosis. And not only are independent assortment and crossing over able to mix up the genetic information within your own individual eggs or sperm, actually creating different eggs and different sperm. Every cell that exists in your body, when we create those, is different. We then talk about the ability of taking just sexual reproduction in and of itself and talking about mating individuals together. And if it truly is random mating, we're mixing up genetic information there again because we're taking two sources and bringing them together. So the genetic variation that comes out of this is just astronomical. And that is going to be the quickest process to allow evolution to function. Certainly, mutations can happen along the way, although they're generally rare. They can provide new genetic variation, but to just take what's already existing in my population, basically throw it into a pot and pull it back out again at random, in and of itself makes plenty of variation in sexually reproducing organisms for evolution to do its job, and that's really pretty cool. So how does it work? So let's talk independent assortment first, okay? So here we're talking about independent assortment, and what that really means is how I line up those chromosomes during metaphase 1. So independent assortment is really going to be important during that metaphase 1 process, okay? And it comes down to how do I line up those tetrads. And the cool thing about it is although those tetrads are going to stack on top of each other, right? What we're looking at here is the way in which that parental information lines up. So here's a really simplistic example. So I've got two pairs of homologous chromosomes, right? Blue ones are from dad, red ones are from mom. In possibility number 1, all of dad's information lined up to the left and split on that direction. All of mom's chromosomes lined up on the right and split that way. So that here are two different combinations that could potentially come out of that original cell. Possibility number 2 is that dad's chromosome 1 went to the left and mom's chromosome 2 went to the left. Mom's chromosome 2 went to the right, dad's chromosome 2 went to the right. So now I'm mixing it up. So now I have a chromosome 1 from dad and a chromosome 2 from mom in these cells, and the opposite, a chromosome 1 from mom and a chromosome 2 from dad in these. So just from independent assortment, in this very simplistic situation, I get 4 different chromosomes, right? When we look at the genetic variation that can come just from independent assortment, you often see it like this, 2 to the n, all right, where n is your haploid number, okay? So if I look at humans, it's 2 to the 23rd, right? We plug that into the calculator just to kind of give you a more round sense of what we're looking at. We're looking at something like this, right? Over 8 million different combinations just in the cells that you can create just because of independent assortment. And then that's not the only way we create different genetic variation, right? There's not only the independent assortment that's happening in your cells, but there's the crossing over that's happening in your cells. And then we talk about random fertilization of it's a random chance which egg and which sperm fertilize, even if you're talking about the same partner over and over and over again for every offspring that you create. So we're talking billions, trillions of different combinations once we encounter not only independent assortment, but take into consideration crossing over, random mating, random fertilization. This is where the variation comes from. So what is crossing over then? So I mentioned this briefly when we were talking about the stages of meiosis. This is going to happen during prophase 1. Okay, so it occurs during meiosis 1, but even more specifically, let's put in here that this is during prophase 1. Okay, so this crossing over is going to happen as a result of those tetrads coming together and synapsing. And what you're going to get is here would be mom's original chromosome, here's dad's original chromosome, and they create these chasms, these sort of crossover components, where I'm actually going to mix up genetic information between the chromosome that mom gave you and the chromosome that dad gave you, and we're going to make recombinant chromosomes out of this. So if I look at how this works, right? So here's my original tetrad, and this goes back to that mouse example of coat color and eye color. Okay, they happen to be pretty far apart on that chromosome. So when I go through, make that tetrad, we're going to create breaks in those genetic codes in the DNA backbone, right? They're going to create that chasm, they're going to split over, line over each other, and then once we seal those backbones back up, they are now mixed up information, right? So dad had originally given you all lowercase e's, capital E's came from mom, same from the coat color, mom gave you the capital C's, dad gave you the lowercase. So when I get down here now, you can see that mom's original capital C is here, but now I've got part of this chromosome being a lowercase e, and the opposite on this one. This is during anaphase 1. We go through anaphase 2 and go through the telophase and the cytokinesis to create those 4 different gametes, right? And they now all have genetic information that is different from each other. I've got 2 chromosomes here that are what we call the parental type, okay? And what that means is that they look exactly like the original parent chromosome, right? So if I look at this chromosome and I come back up top here, right? It looks exactly like mom's. Dad's, same thing. If I were to come all the way back up here, you've got the same combination on that other parental chromosome that came from dad. These two here in the middle are what we call recombinant chromosomes. They're recombined, right? I've got a little bit of mom, a little bit of dad, a little bit of dad, a little bit of mom. And so they're recombining, we're shuffling them up, which again gives us more genetic information. Cool thing about those recombinant chromosomes, this is kind of just a side note here, we can actually use that to map where genes are located on a chromosome. The more likely those genes get separated from each other, the further apart they have to be. So if I had another gene here that was right next to this C, right, and we also went through this, it's probably going to be more likely that those 2 genes always look like a single parent, right? So if I put another chromosome or another letter, let's just say A here, right? And mom gave you the capital A, dad gave you the lowercase a. If I follow that all the way through, all right, for those particular chromosomes at least, or those particular traits, I should say, this should be an A, those always end up looking like a single parent. Hopefully that makes sense, right? So because they're really close together, the likelihood of them separating happens much less frequently. And so for these 2 traits, they would all be parental types because they never got mixed up. But the further apart they are, the more likely that we can create these recombinant chromosomes at a higher frequency.
Hi, it's Mr. Anderson, and in this video I'm going to go through the phases of meiosis. Meiosis is a lot like mitosis. It starts with interphase, but remember, the point of mitosis is to make two identical cells. And in meiosis, what we're trying to do are make four genetically different cells because they're destined to be gametes. They're destined to be sperm and egg. And that's the whole point of sex. We want the next generation to be different than the generation before. And so when you look at a diagram of meiosis, it's a little bit daunting. And we're not going to go through all of it right now. We'll go through this diagram at the end, kind of as a way to review it. But I want to step through each of those phases of meiosis. A quick mnemonic, PMAT times 2 is going to remind you the different phases that we have to go through. So it's prophase, metaphase, anaphase, telophase, and then we go through that again on the second division. Before we get to it, we should talk about the major structures that you're going to see as we go through meiosis. The first one, of course, are the chromosomes. And so you're going to see two of each chromosome at the beginning. And so we get a chromosome 1 from our dad and a chromosome 1 from our mom. We call these homologous chromosomes. Remember, each of these chromosomes has hundreds of genes on it. And so you get two copies of all those genes, one from dad, one from mom. This would be chromosome number 2 because it's shorter. And in my model, I'm just going to use 2 pair of chromosomes. Remember, in a real human cell, we're going to have 23 pair of chromosomes. But it's almost too difficult to follow what's going on if we had that many chromosomes. You just have to multiply it times 13. Now, lots of times you'll see chromosomes not look like that, but like this. And so what's happened here is that we've duplicated that chromosome from dad. And so these two, what are called sister chromatids, are exact copies of the DNA. And so you know when you see a chromosome that looks like this, they've already gone through that duplication. Another important structure is the centrosome. The centrosome is going to organize the spindle, which is essentially dividing the nucleus and also dividing the cell. And in animals, it's made up of two things. We have the centrioles on the middle, and then these microtubules that go around the outside. In plant cells, they're going to lack these centrioles in the middle, and the nuclear membrane is actually organizing a lot of this division. But let's begin at the beginning of interphase. So this is just as the cell has been formed, and you can see right here in the nuclei that we have those 2 pair of chromosomes. Chromosome 1 and chromosome 2. And if we look at the centrosomes, we just have 1 centrosome. And so that's not usually what a cell looks like. This is what a cell usually looks like. And so what's gone on here, you can see that in interphase, we've duplicated those centrosomes. So we have 2 of those. And the DNA is all loose. It's not tightly wound up into these chromosomes that we characteristically see. As we go through interphase, what eventually happens is then we can see those chromosomes again. Now, do you remember what it means when they look like this X? It means that during the S phase of interphase, we've copied the DNA. So we have 2 complete sets of DNA. And this looks identical to mitosis, but it's just about to change. And so what happens next is prophase 1. During prophase 1, the chromosomes undergo what's called synapsis. And so what's happening is chromosome 1 from dad and 1 from mom are coming together, and they're wrapping around each other really, really tightly. And what's really going on is that they're swapping parts of their chromosomes. In other words, these are pretty much identical except for the changes in the genes. And so they undergo what's called crossing over. So segments of chromosome from mom are switching with chromosome from dad. Same thing over here, and same thing with chromosome 2. Now, what's that giving us? It's giving us variation. If this didn't occur, if synapsis didn't occur, the chromosomes that you get from your mom and your dad, you would give to your children, either as a chromosome from your mom or a chromosome from your dad. And what we're doing in crossing over is we're combining the 2 chromosomes that we got from our 2 parents and making a brand new chromosome that we want to deliver to our child. That's the importance of this. If we keep watching, what happens next? We then go into metaphase 1. What's happening in metaphase, you can see that they're all lining up or meeting in the middle of the cell at what's called the metaphase plate. Now, they could have organized themselves like this, with the blue one on the left and the red one on the right, but they could have easily organized themselves like this. So they could have been in a different position. So this would be a totally different orientation of the chromosomes. It also could organize like this for chromosome 2, or it could organize like this. And so what do we have? We have 4 different ways that these chromosomes could orient themselves independently at metaphase 1. What is that giving us? Well, it's giving us variation. And so there are 2 raised to the number of pairs, possibilities of how they could arrange at independent orientation of metaphase 1. So how many are there? We would say 4. What if I had 3 here? Then there would be 8 ways that they could arrange themselves. Still doesn't seem like much variation. But remember, in humans, we have 23 chromosomes. And so there are over 8 million ways that all those chromosomes could independently orient themselves during metaphase 1. And so that's where we're getting, again, that and crossing over is giving us a huge amount of variation in meiosis. And remember that one sperm has to find that one egg. And so it's really over 64 trillion possibilities of an offspring just based on independent orientation itself. Let's keep watching then. So another thing that happens in metaphase 1 is that spindle is going to attach. So you can see that the centrosomes move to either side of the cell, and the spindle attaches to the centromere of each of those homologous chromosomes. During anaphase, it's pulling them apart. So we can see that those homologous chromosomes now are going to either cell. And then during telophase, what's happening is we're reforming a new nuclei at each side. We've divided the nuclei, which is meiosis, but now we have to divide the rest of the cell, and that's called cytokinesis. And so we're done with meiosis 1. We've gone through prophase, where we saw that synapsis. We went through metaphase, where we had that independent orientation. And now we've divided the nuclei into each of those cells, but we're not done yet. So what's going to happen next is we're going to go through prophase 2. Now, during prophase 2, there's no more crossing over, but what's going to happen is those two chromosomes are going to line up. Again, they're going to meet right in the middle, and the spindle is going to attach to each of those centromeres. Now, if we look what happens, each of those chromosomes are being pulled to another side during anaphase 2. And then finally, during telophase and cytokinesis, telophase 2, we've created these 4 cells that we wanted in the beginning. And so if we look at where did we begin, again, way back in the beginning, we had 4 chromosomes, or 2 pair of chromosomes. Now we have 4 cells, and each of those only have 2 chromosomes, a 1 and a 2. And so what would happen next in the circle of life? These would fertilize a sperm, or excuse me, if these were sperm, they would fertilize an egg, and then we'd start over again. We'd have a brand new organism that's going to be created through mitosis. Now, this is how we make the sperm. Each of the sperm are going to be made like this. Again, there'd be way more chromosomes in us because we have 23. It's a little different with the eggs because there's so much important material in the cell that only one of these nuclei will actually be used and the other ones won't be used genetically in that cell. And that allows us to keep all the important parts of the cell, like the mitochondria, endoplasmic reticulum, in that one cell. And so now let's kind of review and go over that confusing diagram at the beginning. So what are we looking at here? This would be interphase at the beginning. You can see we just have one copy of all those chromosomes. This would be at the end, so you can see that we've duplicated the DNA. Next, what do we have? This is prophase 1. To remember what important thing is occurring there. We've got crossing over, and then we've got independent orientation during metaphase 1. They then are pulled apart, and then we have 2 cells. And now in the second meiosis, what are we doing? Is we're just lining up those chromosomes, and then they're splitting up into each of the sides. And so what do we get at the end? Each of those cells, if we were to go back to here, each of those cells, look at this one, and that one, and that one, and that one, are totally different than that original cell. They also have half of the DNA that original cell did, and so that's meiosis, and I hope that was helpful.